Welcome everyone. After Zoom had its regular reminder. Well, here we go again. Welcome everyone. I'm Sangeetha Iyer. I'm ACD Scientific Advisor, and I am here today with ACD's Executive Director, Heidi Wallace. Heidi, say hi.
Hello.
And Dr. Steve Baker, who I'm very, very excited to introduce. So Dr. Baker is a clinical pathologist at University of Utah, and he recently presented ACD with a very exciting proposal to research a potential treatment approach for CTD and to set up the very first CCDS Research Center with full-time dedicated staff to work on translational research for both CTD and GAMT. Welcome, Dr. Baker, and thank you for meeting with us.
Hey, Sangeetha, thanks for having me. This is truly an honor and a pleasure to be here, and I'm so happy to meet with the community and have this webinar. So thank you.
It's entirely our pleasure. So I'm going to, if it's okay with you, I'm going to go ahead and address you as Steve for the rest of our interview.
Please, yeah. Thank you.
So we can kick things off in a sort of
if I can call you
manner.
Dr. Iyer.
All right, so I think what we are here to do today is talk a little bit about cerebral creatine deficiencies and sort of get a sense of your interest, how you got involved in this research, and what you think is important as we move towards translational cures for CCDS. So Heidi and I will both be going through questions that are of interest to us and we think of interest to the community, and we'll give you a chance to dive into the details.
Great, that sounds perfect. Yeah.
Awesome. So I guess I'll get started first. How did you learn about creatine deficiencies and specifically CTD?
Yeah, so I guess in a way of giving a little bit of background, so I did my PhD in pediatric neurological disorders. So I did my MD PhD training at Baylor College of Medicine, and I was a graduate student of Huda Zogby. And so Huda is kind of known in the field for having discovered the gene that causes Rett syndrome. So that mutations in MECP2 cause Rett syndrome, which is a neurologic disorder that affects primarily females because it's an X-linked disorder, and males are kind of more severely affected and generally don't present with this sort of milder form that presents in females. And so I have always had an interest in helping children with neurologic disorders. And then when I was a postdoctoral fellow at Stanford, I did my postdoctoral fellowship in the lab of Tom Montine, and he is very interested in creatine and how creatine is metabolized in the brain. And one thing that I was passionate about was helping children with neurologic disorders, and it just so happened that I learned about CCDS, so Cerebral Creatine Deficiency Syndrome, and wanted to understand how creatine and the lack of creatine causes that disorder. And so we explored the sort of expression of the synthetic enzymes in the creatine synthesis pathway, particularly within the brain. And that's where I realized that there might be an opportunity to help ameliorate the majority of one of these particular subtypes of CCDS. And so that's kind of what sparked my interest in this project. Yeah.
That's great. So can you share a little bit about your basic goals and concept that you are wanting to pursue in this project and how it relates to those findings?
Definitely. Yeah. So one thing we found was that, there's this traditional view of creatine metabolism where creatine is synthesized. And I have some slides up on the screen just as talking points. But so kind of the traditional view is that creatine is synthesized in the body using a cooperation between two organs. So about half of the body's creatine or daily needs for creatine is synthesized endogenously. The other half is, of course, obtained from the diet. And it's thought that the kidney has the highest level of AGAT expression, so the first synthetic enzyme in the pathway. And then the liver has the highest expression of GAMT. And so it was originally kind of hypothesized, decades ago, based on the expression and activity of these enzymes in these two organs, that essentially the kidney synthesizes an intermediate, which is GAA, or guanidino acetate, using two amino acids, arginine and glycine. And then the liver finishes the job by basically just methylating that GAA into creatine. And then that creatine is supplied to the rest of the body. However, one thing that we discovered when we looked for the expression of these two enzymes was that the story wasn't quite that simple. So if you look, and forgive me if I'm getting a little too technical or in the weeds here, but essentially these colorful images are just representations of where these enzymes are. And we use other markers to help us see which cell types are lining up with the expression of the enzymes. And so one thing we noted is traditionally it was thought that the liver expresses GAMT, but not AGAT. And so AGAT was thought to be mostly in the kidney, but we actually, when we stained for AGAT, we detected AGAT within the liver, but it wasn't in the main cell type that is there. So the liver is primarily made of hepatocytes. And those hepatocytes, indeed, like the traditional view suggested, express a ton of GAMT. So here, this red stain is all GAMT. And you can see that it lines up with this marker for the hepatocytes, albumin. But then there's these little dots of AGAT that we could detect, and they actually lined up with this marker from the endothelium, but not albumin, the marker for the hepatocytes. So the blood vessels that penetrate into the liver and feed the liver with nutrients are lined by these endothelial cells. And it turns out that they're the ones who express AGAT in the liver. So it's the picture wasn't just so simple between the kidney and the liver, where these two enzymes were separated in different organs. And just to show you what that looks like in 3 dimensions, so this is a confocal microscope image of a liver from a mouse that we fixed and stained for AGAT, so the first synthetic enzyme in the pathway. And you can see these little red speckles kind of flicker on and off. And we co-stain with albumin here to show you that they're just on the outside of the albumin signal. They're not within the hepatocytes themselves. But when you overlay it on top with the endothelial cell marker, you can see that actually those red dots are overlaid by the purple staining here. And so to us, that showed us that, in fact, the liver does express both enzymes, and that the AGAT signal is coming from the endothelium and the GAMT signal is coming from the hepatocytes. And so we wanted to ask, where in the brain are these enzymes being produced? Because some earlier work from Olivier Bressant's laboratory showed that these enzymes are expressed in the brain. But that was back before the era of single cell RNA sequencing technology. And so we wanted to look into these new larger data sets of single cell RNA-seq and ask where are these two synthetic enzymes expressed. And again, I try to avoid getting too much in the weeds, but suffice it to say that oligodendrocytes are kind of the king of creatine synthesis. So compared to all other cell types in the body, oligodendrocytes express more AGAT, which the gene name for AGAT is GATM, and they express more GAMT than any other cell in the body. So and what we found in the liver was true at the RNA level, where the hepatocytes express GAMT, but the endothelial cells in the liver express AGAT or GATM. And so this told us that the RNA-seq data that we were looking at was reproducing what we were seeing at the protein level. And it really suggested to us that oligodendrocytes are producing creatine more than any cell type in the body, at least at the synthetic enzyme level. And so we wanted to see, where in the brain these enzymes are being expressed. And in fact, it mapped perfectly to the RNA-seq data. So here is a section of mouse brain It's kind of spinning ad nauseum, but you can see the red signal is coming, is right outside of these blue nuclei that are in the white matter tracts. And white matter tracts are where oligodendrocytes live. And so we, just like the single cell RNA-seq data showed us, we were detecting the first synthetic enzyme in oligodendrocytes. And the same was true for the second synthetic enzyme. And I'll show you another image of that. So this is the second synthetic enzyme, and it is also being found in these white matter tracts in oligodendrocytes. But unfortunately, we didn't really see it being expressed in neurons. So the guys that are stained in green are the neurons. So the first synthetic enzyme, I should say, is only in oligodendrocytes and is not in neurons. And so that suggested to us that what we could do was, as a potential therapy for creatine deficiency, is that we could potentially get the neuron to synthesize creatine on its own. And so how do you do that? So if the neuron naturally expresses the second synthetic enzyme in the pathway, but it doesn't express the first synthetic enzyme, maybe we could make the neurons express the first synthetic enzyme in the pathway to then complete the link and get the neurons to be able to synthesize creatine entirely internally. And so that is the overarching goal of this proposal, is based on the single cell RNA sequencing data and then the immunofluorescence, where we were able to detect the second synthetic enzyme in neurons, but not the first synthetic enzyme. Can we complete the creatine synthetic pathway within neurons, which ordinarily exists entirely in oligodendrocytes?
Wow, Steve, thank you so much for walking us through that. That was brilliant to see both the RNA-seq data and how that lines up with the tissue observations, brain tissue observations. I guess I have a couple more questions on what you just presented. So I'll tell you my takeaways, and I'll have you fill in the gaps. So I think when you started off, you said that we get 50% of our creatine from diet and 50% of it synthesized in our bodies. So for people in this community, if they're not able to synthesize creatine de novo, why is it that they just can't take a large quantity of creatine and have that sort of supplant the rest?
Absolutely. Yeah, no, that's a great question. In fact, so, CCDS is broken up into 3 subtypes. And I think I have a slide to represent those, or maybe okay. So, well, I don't have my slide here, but suffice it to say, there's CCDS type 1, which is owes to the lack of the transporter. So it's a mutation in SLC6A8, which encodes the creatine transporter. And then there's CCDS2, right? And that CCDS2 is also known as GAMT deficiency. And then there's CCDS3, which is rarer, but owes to a lack of a functional AGAT. So if you have a defect in any of these 3 components, then the resulting phenotype is very similar, which is CCDS. And so in patients who have GAMT deficiency and AGAT deficiency, if you intervene early enough, and usually the intervention is dietary, by supplementing creatine and occasionally some of the precursors in the diet, then the body can actually use the transporter to take up creatine into the cell, and it can be it can function. And athletes use this pathway all the time to achieve muscle hypertrophy, right? Because we know that creatine is very important for the brain, but it's also important for muscle function and enhances muscle function. And in fact, the places where creatine is the highest is in skeletal muscle and then also in the brain. And so you can, in the case where you have a defect in the synthetic enzymes, supplement creatine through the diet to make up, at least partially, for what the body cannot produce because you have a defect in the synthetic enzymes. But if you're missing the transporter, well, then the problem is that you could, in theory, try to supplement through the diet, but the body could not take up that creatine either through the gastrointestinal tract or into the cell. And so that has been the longstanding problem that is preventing us from treating patients with CCDS type 1, which is, of course, the most common type. About 70% of CCDS is CCDS type 1, which is also called creatine transporter deficiency, or CTD. And so, unfortunately, because of the lack of that transporter, supplementing through the diet doesn't seem to benefit those patients. And so the hope is, how can we circumvent the transporter? And the reason why the transporter is so important is because it gets creatine into the cell from the outside. So some other cell type or the diet is providing the creatine, but it can't get into the cell. Well, how can we bypass that cellular transporter? And the way to do it, my hypothesis is that if you can get the cell to synthesize creatine internally, then you do not need the transporter. And that's the idea, is that in these patients who have CTD, they don't have a functioning transporter, or they have a partial functioning or only weakly functioning transporter. Let's get we know that neurons are likely the cell type in the brain that require creatine. The reason we know that is because they have high levels of expression of a particular type of creatine kinase that is indicative of cells that require creatine. So let's get the neurons to express the first synthetic enzyme in conjunction with the fact that they endogenously produce the second synthetic enzyme, and therefore they can bypass that need for that transporter.
Very cool. So what I'm understanding is that this is in particular the case for resolving the creatine deficit in the brain, because in the brain where I've understood that creatine is particularly critical in developmental stages as well as you go on with life, that's the cell type that we do need to find a way to replenish creatine in. In the periphery, there are other maybe cell types where you have AGAT and GAMT sort of in slightly separated positions, but potentially they can still be some synthesis of creatine. But in the brain, you don't have neurons, you don't have cell types that have AGAT and GAMT in the same cell type, and particularly close to the neurons, which is where you want to reintroduce creatine.
Yeah.
Go ahead.
No, that's exactly right. Yeah. So like this cartoon model that we sort of have worked up, so exactly right. So in the brain, so this is that creatine kinase that I talked about, is creatine kinase mitochondrial 1. So CKMT1 is expressed exclusively in neurons, and you can see that here by the purple coloring. And then down here, they label the different cell types that are found in the brain, and these are the neurons, which are these blue ones. So we know that they express the creatine kinase, and creatine kinase is what lets cells use creatine in order to buffer for energy, and it's the role that creatine is hypothesized to play inside the cell. And so we know that the oligodendrocytes here, just showing you a blow up, express both synthetic enzymes, and somehow they're able to generate, we think, creatine and then export it through the CSF or directly to the neurons. And then the neurons require CT1 or the creatine transporter to take it up. And so exactly like you said, the neurons unfortunately do not express both synthetic enzymes. And in fact, they're probably relying on these oligodendrocytes, which do, to get the creatine inside the cell, or to be the source of the creatine, which is then taken up by CT1 into the cell. But if we can get the neurons to express both synthetic enzymes, then they will no longer be as dependent on the oligodendrocyte for that synthesis of creatine. Yeah.
That's excellent. And so what you're proposing is gene therapy, or reintroducing a gene, or and using that to sort of reintroduce an enzyme or a protein that is going to be essential. But this is a differentiated approach from doing gene therapy for SLC6A8 or the transporter itself. You are trying to reintroduce something else that is important for the de novo synthesis of creatine.
That's right. And so other laboratories have proposed replacing. So if 70% of CCDS is due to mutations in SLC6A8, which encodes the creatine transporter. So one very logical and important mechanism to replace the function of that transporter could be delivering the gene that encodes the transporter itself, deliver a normal copy of SLC6A8 in order to replace CT1 in patients who have a defect in that gene. The trouble with that is twofold, I think. And so the first is that the vast majority of patients who have mutations in SLC6A8 have truncating mutations. So here I'm just showing you a pie chart of the different types of mutations in SLC6A8 that are annotated in ClinVar that lead to CTD. And so the bigger slice of this pie, the more than 50% of this pie, are patients who either have a large deletion, they have what's called a frame shift mutation, or a nonsense mutation. And when you have one of those mutation types, what happens is you generally do not produce the full protein. So you produce only a piece of the protein or no protein at all. And so if you then try to reintroduce the functioning copy of SLC6A8 into their brain, their body will have never produced that protein. And so it could potentially see it as a foreign antigen and mount an immune response against it. So that's one thing that we see when we try to do enzyme replacement therapy, for example, in patients who have hemophilia A. So I'm a blood doctor by training, and one of the diseases that we treat are patients who have a complete loss of function in a gene called factor VIII, which is part of your body's natural ability to clot. And when we try to supply factor VIII to patients who have factor VIII deficiency, their body occasionally mounts an immune response against that as a foreign antigen. And so there's the potential for that here. If we try to reintroduce this gene into the body of a patient who has never ever produced the full length protein, they could potentially recognize that as a foreign antigen. Whereas in contrast, if we try to supply AGAT in patients who have CTD, they do not produce the transporter, but they do naturally produce AGAT. It's just not in the right cell type that we need it to be in order for the cell to internally synthesize creatine. So we could supply AGAT, and that would be an endogenous antigen for the body. It would not see that as a foreign antigen. And so that would circumvent that potential problem. The other potential problem is that if you look at the single cell RNA-seq expression of the transporter, so the transporter, just showing you these purple dots, is sort of kind of scattered expression throughout the brain. And one of the limitations of gene therapy is that generally when you deliver a gene by gene therapy, you have to express it under the control of a promoter. And expressing it in the right cell type and in the right cell context can be very difficult with a single promoter that is not the endogenous full-length control element that a gene is normally expressed in. So there's the potential to sort of express the transporter either in a cell type that is not perfect for it, or in the context where it's not perfect for it. And all I'll say is that the creatine transporter is an electrogenic channel. So the way it takes creatine into the cell is that it incorporates 2 sodium ions and 1 chloride ion in order to pump creatine inside the cell. And in so doing, if you just balance out the charges, creatine itself is a neutral molecule. It has a formal positive charge and a formal negative charge, which balance out at body pH. But the sodium ion is positively charged, and the chloride ion is negatively charged. And therefore, this transporter, when it transports in creatine, because it transports 2 sodiums, 1 chloride, and then 1 molecule of creatine, it generates a net positive charge when it moves into the cell. And so if you express this in a cell type that is not ready for it, or in an amount that is not perfectly tailored to the endogenous control, there's the potential that you can cause changes in the membrane potential. And that may be part of what's causing some toxicity when we try to re-express this channel in mouse models.
That's great. Thank you. I learned so much every time I talk to you. As a parent, I think I'm going to rephrase some of this and just make sure that I've understood you correctly. The thought is that the body is accustomed to and not going to have a negative response to AGAT because there is AGAT throughout the body. So introducing it inside of these neurons where there is GAMT, and now there would be AGAT, we would now have a creatine factory within the neuron where it is needed. And so the fact that the transporter doesn't work to bring in creatine is no longer an issue. We've got it where we need it. Can you explain how you would pursue this in the research center and kind of what your plans are there?
Absolutely. So thanks so much, Heidi. Yeah, and I learned much more from the community than you will ever learn from me, I promise you. Patients are our best friends and advocates, and patient families. So thank you all for being there for us as researchers. So yeah, the goal is let's leverage the expertise that we have here at the University of Utah in order to build a center of excellence in order to see this potential avenue for therapy through. One thing that we would need is we would need to test this model in cell culture. And so the hope is, so we here at the U, so we're lucky to have Marzia Pasquale and Nicola Longo, who are both longstanding and really well-respected and knowledgeable people working in the field of creatine deficiencies. Nicola actually sees patients with creatine deficiency, and Marzia is an expert at mass spectrometry. And those together, the expertise that we have here at the University of Utah would allow us to test this model in cell culture. And the way that we imagine doing this would be to take neurons. So let's take neurons that are deficient in the creatine transporter. So luckily there is 2 mouse models for CTD. And these recapitulate many of the phenotypes that, or at least neurological symptoms that we see in patients who are affected by CTD. And so what we can do is we could make cultured neurons from either the wild type or the loss of function of CT1, and then try to deliver AGAT using a lentivirus, which is just a viral vector that people use in gene therapy, and to deliver a gene payload, which is AGAT, and express it within these cultured neurons to see if they can then start to synthesize their own creatine internally without requiring the transporter. The other thing that we're working on is there are protocols for differentiating human neurons from iPSCs. And luckily, the Coriell Institute has lines of human iPSCs that are deficient for a functioning SLC6A8 gene. So we can request those iPSCs, and sorry, I'm probably getting in the weeds here, but induced induced pluripotent stem cells that are derived from a human patient. So what happens is they take a fibroblast from somebody who has CTD. They use a set of genetic tricks to trick that cell to become like a stem cell, almost like an embryonic stem cell. And then what we can do is we can vary the culture conditions. We can keep them in a plate just like this and add the right factors to that plate, and we can get them to differentiate into neurons. And then those neurons are just like the neurons that would be inside of the brain of a patient with CTD. And so the hope is that we could use the fibroblasts that are generated by donations from the community to make these induced pluripotent stem cells, differentiate them into neurons, and that would give us a human neuron substrate to work with and do our experiments to see if we can express the first synthetic enzyme using a lentiviral transduction system and get those human neurons to produce a creatine internally without requiring the transporter. And so those are essentially the two strategies that we've proposed to perform in this grant. Is can we first test this in mice? And I think that's important because ultimately, before we went into real in vivo human clinical trials, we would have to show preclinical data to the FDA that this would work in a mouse model. And the simplest format is using it first in cell culture, but then ultimately progressing that into in vivo models. So inside of a mouse, a living mouse with a defect in the creatine transporter. But then also in parallel, performing these experiments using human neurons that we derive in a safe way without having to take human brain biopsies or get primary cells by taking fibroblasts from the skin and differentiating those using this genetic trick into human neurons in a dish, and then seeing if this therapy works in that setting as well.
That's great. I think it's so, it'll be amazing to see this come to fruition because you'll be validating, proof of concept therapy in in human relevant cell systems as well as in a multicellular sort of more complex mouse model system. So I guess the perfect segue to trying to understand what kind of resources will be necessary to accomplish this work.
Yeah, so, okay. So this is a mock timeline, and we are sort of progressing through this first quarter of the year now. So in essence, we would need essentially 2 full-time technicians with laboratory supplies and essentially 2 years. And so what we would do in the initial portion of the funding proposal period would be to acquire the reagents that we need. So one thing that we need to do is get the mutant mice that have this defect in the creatine transporter. And then we need to acquire the plasmids that we need to make the lentiviral vectors to express AGAT in neurons that we ultimately derive from those mice. And of course, we'll have to hire the staff to perform these experiments. And then we will have to do what's called molecular cloning, which is you take the AGAT gene, you use kind of DNA scissors to cut it out, and you put it into the lentiviral vector. And then you establish your founding mice to start a breeding colony. And at the same time, we would contact the Coriell Institute and get the human pluripotent stem cells that we need to also make human neurons. And then we will have to keep those cultures and the mice in breeding for the remainder of the 2 years. So that will incur some costs. So the initial purchase of the mice is a few $100s. The cost of a mouse cage per day here at the University of Utah is actually pretty affordable relative to the several institutions in the nation. So it's $1 a day that we need for each cage of mice. And so we are estimating that we'll need about 10 cages of mice to generate the raw material to do the experiments. So then as we progress through the first year, we will start to generate the lentivirus that we've completed by cloning. And then we will begin the experiments that I discussed, which is take those lentiviruses that we make with AGAT, deliver them to neurons which are deficient in the transporter, both human and mouse, and then analyze them by, do we see AGAT expression? Do we see endogenous creatine synthesis? And is that creatine synthesis at a level that is sufficient to reproduce what would normally be there in a normal wild type, in a normal healthy neuron? And so those are critical questions that we need to address in order to decide whether this is a viable strategy for long-term treatment of patients. And then at the same time, I'll leave the details out here, but there's always another alternative for gene therapy. Instead of delivering the gene in total, there are newer technologies like CRISPR. And one of the things that we've proposed to do is, instead of delivering AGAT itself, so instead of delivering the gene in total, what if we turn the neuron in, the neuron's AGAT gene on itself? And one way to do that is using CRISPR technology. And so that's what we've kind of outlined in the later part of the second year. So the first part is, let's deliver AGAT. Let's see if we can get neurons to bypass the need for the transporter, both human and mouse neurons. And then the second part is, okay, what if we can, instead of delivering the whole gene, we can use CRISPR to modify the AGAT locus to naturally turn it on within the cell to produce AGAT within neurons. And so those are the kind of the two main spearheads of the proposal that we've written to work out in the next 2 years, and the resources that we would need. So.
That's fantastic. Thank you. Thank you so much for sharing. I think as we wrap up, in our community hears about this project that's very unique and exciting, there are probably questions of how can we help? And I will quickly just summarize that from ACD's perspective. As you mentioned, you'll be using samples from Coriell, and we are we're here to help the community connect with Coriell to donate those samples. And so definitely reach out to ACD if you need instructions and support there. Help, join our Holiday Heroes promotion and help raise the funds that ACD needs to support this work. And then, of course, participate in our patient registry so that we can continue to collect data about the progression of the disease, which will be relevant in a hopeful trial down the road to establish endpoints and whatnot relative to the disease. So thank you, Dr. Baker and Sangeetha, for joining us today. And as we've heard, Dr. Baker has an exciting hypothesis and a strong plan to move forward. And that research is taking a new approach to find a way to treat CTD. So we're currently in the midst of the Holiday Heroes campaign. I think today's interview with Dr. Baker proves the point that, this year our campaign is very different and very pointed at an important and promising project. And so we've set a lofty goal of raising $250,000 this year, but we feel it's achievable, and we've made great progress so far. And we have some matching donors that we'll be announcing soon to help us cross that line. So we hope everyone will donate and share this information with their friends and family and help us make real progress. And we will have a recording of today's conversation available very soon, so you can share that as well. So thank you again very much for your time, Dr. Baker, and happy holidays, everybody.
Great. Thank you, Heidi. Thanks, Sangeetha. This was awesome. So, and thanks for, being such an amazing supportive community. It really helps researchers out. So we appreciate you guys.
Thanks, everyone. Happy holidays.